episode 292 of the Stem Cell Podcast, Biomedical Ethics, with Professor Hank Greely. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Hank Greely. He's from Stanford University, and he's on the podcast to talk about his work on the biomedical ethics of genetics, neuroscience, stem cell research. We're probably going to go even farther afield, but he's thought about all of it. We've also got our usual roundup of recent highlights and stem cell news coming right up.
But first, we're just 2 months out from ICCR 2025, and we can't wait to meet you all in Hong Kong at the Global Stem Cell event happening June 11th to 14th. Register and check out the program at iccr2025.org.
I know who we're going to see at that meeting. I hope to see him there. We've seen him at every meeting we've been to in the last few years. Always grab a coffee or something, talk about his next 5 or 6 papers that he's got cooking. And that's Sergiu Pasca, who I'm going to lead the roundup today with a story out of his lab that is once again resetting the bar for assembloids and, really extending the ambitions and applications of organoid biology. In this case, we're talking about the ascending somatosensory pathway, right? Which conveys environmental and bodily sensory information from peripheral organs to the central nervous system, right? So these somatosensory neurons, their cell bodies are located in the dorsal root ganglia and trigeminal ganglia. And they innervate peripheral organs, including the skin, in order to, as I said, detect these somatosensory signals and then to transmit them. And this is key. They transmit them to the spinal cord and to the hindbrain, right? And then there's other neurons in the dorsal spinal cord or the hindbrain that then transmit that information to other brain structures, namely the thalamus is one of them, right? And then the thalamus can relay these signals to the cerebral cortex, right? And that's how the signals are integrated. So there are 4 nodes on that signal chain there. Just a note, we're going to come back to that. But the way that the ascending sensory pathways form during human development, and key here is the way that functional defects arise in that developmental process that can result in pathological conditions. It's not understood, not known, and obviously because it's the brain, right? It's not accessible, particularly during development. And there's not animal models that can recapitulate the physiology or electrical signal conduction, all the different cell types, all the intricacy of the human brain. And even if they could, it's not really accessible. You can't really see in real time, right? It's still a black box. You can't get into the brain. So, the lead here is obvious, going up to why we can use organoids and assembloids to recapitulate the process. But I really don't think there's another system that is more well suited to really ambitious applications like the one I'm about to describe than the brain, because there's all these different parts, right? And the rationale for being able to monitor that signal in real time, as I just kind of alluded to, it's there's no other way, right? So you know where I'm going with this. This previous work from Sergiu's lab had developed the 3-part assembloid. We talked about that on the show, and that at the time, of course, was revolutionary. No one had ever done 3, and that was to recapitulate this descending corticospinal muscular signaling, right? But why stop at 3 organoids in sequence when you can do 4? And that's what was achieved here in this Nature article, which has been a long time coming. It's this 4-part assembloid that integrates somatosensory, spinal, thalamic, and cortical organoids, right? And it works. I mean, that's the bottom line. It works, which is an incredible achievement. And it was proven to work this circuit by transcriptomic profiling on one level that showed that all the key cell types were present in the circuit, and then real-time imaging that showed the actual connection between the dorsal spinal cord neurons and then further connection to the thalamic neurons. It showed there was a coordinated synchronized response to noxious chemical stimulation. And this is what was so cool to me. Of course, the application, why this is an article, uh, it, they did some modeling, of course, which was super cool. First, they modeled this loss of this channel, sodium 1.7 channel, which pathologically causes insensitivity to pain, which may sound like a superpower, but turns out that it sucks. And when they modeled that, they disrupted that synchronized signaling relay in these four-part, what are they calling, the ascending somatosensory assembloids. And then the converse they also did. They did a gain of function of this SCN9A variant, which is associated with extreme pain disorder, which is definitely not a superpower. And they found that they were like hyper synchronous. So this story, like all the stories that come out of the Pasca lab, it really covered all the bases in terms of an ambitious model, just an achievement in terms of the cellular biology and engineering, and modeling, creating a brand new model of brain function and connectivity, and then demonstrating the utility here in these pathological contexts that no one really even thinks to look at, because they seem so impenetrable. But here, I don't know, creating a model where you throw some drugs at it, right? Or maybe some other kind of therapeutic, cellular, whatever it may be, to try and mitigate the symptoms associated with that pathology or reverse it. So a really complete story as we are accustomed to from the doctor. Nice work, my man.
Yeah, good stuff coming from Sergio's lab as usual. Yeah, I mean, this is perhaps one of the most complex forms of assembloids created to date. I mean, even so, they're still somewhat immature, right? They represent, of course, an early phase of fetal development, as is the case with anything pluripotent stem cell derived. And I mean, perhaps a more immediate use, as opposed to the study of the pain, would be, studying neurodevelopmental disorders, autism, something you're alluding to. Folks with autism have hypersensitivity to pain, and are very sensitive to sensory stimulation, as we know. And so this could be a really useful system for studying that. There's a lot of other neural pathways that I think are also desired to be recapitulated and reproduced in this model system. So there's a lot of things you can do to improve the model, as well as like the specificity of the connectivity across the different organoids that are being put together in the assembloids. But regardless of the limitations, just an exceptionally powerful model system to study this neural connectivity across different subtypes of organoids. So, shout it again to the millionth time to Sergio and his lab for this really exceptional work.
Yeah, it's a great story. And all the applications aside, potential or real, whatever. I think just the ambition, it sounds dumb, but like, yeah, four organoids connected. I like to think of the work that comes out of the lab, and Sergio himself is kind of like the flag bearer at the vanguard, right? He's holding it up so we can all look ahead and see where we're going. And this encourages me. I mean, I'm not the rationale for using assembloids in my work is, well, it's there, honestly, and I'm looking into it, but the complexity, like, I don't know that I need to put four of these things together. But I think exploring the outer limits here in a system where it actually makes sense, the brain, and where connectivity can be manifest and demonstrable, I think it makes me believe that any other combination of cells and other systems is also at least worth attempting, and could have a real benefit in terms of recapitulating something that's more physiological, whether it's to model something or to generate an output in terms of cells. that's more useful. So I really appreciate you, Sergio, for holding the flag and leading the field.
Absolutely. And like what you alluded to, I think Sergio, as being that flag bearer for assembloids, has broadened the utility and the use of these models across stem cell biology. And that's beyond just neuroscience. I mean, I know folks in cardiac biology who are developing their own, quote, assembloids, looking at different subtypes of cardiac organoids, being able to fuse them together, fuse them with vascular organoids, and so on. So I think the power of these models extends beyond neuroscience and modeling, for example, autism and pain and all these other phenotypes associated with neurodevelopment. It's very broadly applicable here, but we're going to stick in the brain. We're going to stick with neuroscience for the next paper that I'm going to be talking about here. Cell stem cell paper titled "A Cloaked Human Stem Cell Derived Neural Graft Capable of Functional Integration and Immune Evasion in Rodent Models." First author here is Chiara Pavan, and this is coming from the lab of Claire Parrish down under, down there at the Florey Institute of Neuroscience and Mental Health in Melbourne, Australia. When I think of cloaking of pluripotent stem cells, you know where I'm going with this. The first name that comes up is Sonia Schrepfer, who has done a ton of work in this area at UCSF and Sana as well recently. She actually is moving down here in my neck of the woods at Cedars-Sinai. So she's going to be part-time down here at Cedars as well. So it's been great catching up with her in some of our faculty meetings as recently as last week. But this is not a Sonia Schrepfer story. This is, like I said, coming from the lab of Claire Parrish. But the concept is pretty similar, right? Pluripotent stem cell derived therapies are all the rage these days. We've covered a lot of papers and talked to a lot of folks who are working in this area. Realistic possibility for a ton of different disorders, including Parkinson's. We've talked to Blue Rock, we've talked to other folks in the field who are working in this area as well. And while those folks are often geared towards generating replacement neurons, you really have to deal with the other side of the therapy, which is immunosuppression, the immune system, rejection, all this kind of stuff. So oftentimes you have to deal with these immunosuppressive drug challenges to actually prevent prevent rejection of whatever therapy you're working on, even if it's not in the brain, of course, right? So here they developed a custom human pluripotent stem cell line, which they turned H1FS8IM. Not the catchiest name, I gotta say. But it was engineered to overexpress 8 different immunomodulatory transgenes to actually enable transplant immune evasion. So there's your cloaking right there. That's what they called it, quote, cloaking, right? At first, the co-culture system, where this custom pluripotent stem cell line, which I'm just going to call 8IM, the neurons derived from 8IM, these midbrain neurons, were able to evade rejection by T lymphocytes in vitro, like I said, but not just T lymphocytes, NK cells, natural killer cells, also macrophages, dendritic cells, all the different populations, immune populations in vitro. They were able to, quote, cloak themselves from being eaten up by those immune cells. Next, they moved it to the in vivo, of course. This is a cell stem cell paper, so you probably got to do that. And then they looked at this 8IM line in humanized mice, where they generated allogeneic neural grafts and also were able to evade rejection in those humanized mice, while, of course, the control pluripotent stem cell derived neural grafts that were not derived from this custom line evoked, as you might expect, activation of various types of immune cells, elevated inflammatory cytokines, ultimately leading to rejection and causing spleen and lymph node enlargement. So there's your control, there's your in vivo and in vitro experiments showing that this custom pluripotent stem cell line creates neurons that are able to be shielded from the host immune system. And then going a little bit further into the functionality of those grafts that were in the humanized mice, they retain functionality and it was more than just they were shielded from the immune system. They actually improved motor function in the Parkinsonian rats. So the additional, and they took it actually one step further. I think this is really important for clinical translation, potentially the incorporation of this quote suicide gene, which allows you to wipe out populations that are not good, that are over proliferative, whatever, right, in those grafts, which is great because, of course, you want those terminally differentiated neurons. You don't want to have any sort of residual pluripotent stem cell population, and you can wipe it out with this quote suicide gene. So this is great. I think this is another tool in the toolkit, weapon in the arsenal, whatever you want to call it, for generating immune evasive stem cell derived therapies. Focus really here is on Parkinson's and generating, immune cloaked neurons and midbrain neurons and dopaminergic neurons. And ultimately, yeah, the next step is going more towards clinical trials. Hopefully you can generate this population wide, applicable, safe, off the shelf cell product where you can envision that this line could be not just used for treating Parkinson's or neurodegenerative disease. Maybe you could be generating cardiomyocytes or whatever for incorporating into those cell therapies as well. So I think a really useful tool for a broad range of applications here.
Yeah, this you led with it. When I saw you shared the title here, I thought this was a Schrefer story, and I wouldn't say they were ahead, but they're probably ahead if we're honest. And maybe it doesn't matter, as you said. It's good to have multiple approaches. I don't think I appreciate the science well enough to speak to whether or not this is like a hedge or a different, fundamentally different approach from the Schrefer approach. Maybe you could speak to that, but either way, I'm fascinated by these immune cloak, whatever you want to call it, stories, because I think they really do represent the solution, a scalable, practical solution. In that it's off the shelf. I think it's fascinating, though, this idea of the suicide gene and as a safety hedge, because it just seems so risky, or I don't know, strange to claim safety with the cell that integrates and functions, and then like you pull the ripcord. Like I wouldn't want to be that patient. But I'd be fascinated to see how that manifests, how you have like a integrated graft, and then because it depends on the disease condition, right? There's a lot of things where it really wouldn't matter. I don't know, like T cell targeting cancer, for example. Like, yeah, okay, you pull the ripcord. A cardiac graft, by contrast, yeah, probably not so much. So I think that maybe I'd be interested to see how they take this story moving forward, maybe into a larger animal model, maybe using something more sophisticated, looking at the degree of incorporation of the graft and longevity of the graft. So when I say that the Schrefer group's ahead, I don't know that they are necessarily at a not competitive level, but I do think that the tech there has matured to the point where they're really applying it at these more preclinical questions, I would argue, than what we see here. But still, I love to see another tool, like you said.
Yeah, I think there's a lot of folks working in this area. It's not just Sonia's group. It's not just this group. I think the folks that are probably the furthest ahead when it comes to these quote immunocompatible approaches are the folks who are generating those custom pig hearts,
Mm.
right? With all the edits that make them immunocompatible for human transplantation. I mean, those have already gone into people, multiple people. So they are probably the furthest ahead. But like we're both talking about, it's good to have multiple avenues to the same endpoint here.
Yes. And I think this is evidence that we're going to start to see it with a lot more disease indications, which I think is the most exciting thing, is how many problems would be solved by having an off-the-shelf product, which is probably why it's so many people into it. So for the last story here, I have something that's perhaps a bit out of left field, but I really love the narrative here. And it's a really important fundamental observation that was made, but it's how it was made that I find so fascinating and interesting. So it's a story about neural. I'm staying in the neural realm here. 3 out of 4 for us today are going to be neural. And it's about neural stem cells, right? So just a little orientation. At the beginning of neurogenesis, you have these neuroepithelial cells that are in the neural tube, and uh, they represent these earliest neural stem cells, right? Which then become or generate radial glial cells, And those are the ones that differentiate into neurons and glia, right? In the adult brain, neural stem cells exist pretty much in two observed regions, which is the subgranular zone and the dentate gyrus of the hippocampus, and then in the subventricular zone, which is right by the lateral ventricles. And in those regions, you get neurogenesis that persists throughout postnatal life, famously demonstrated with these carbon-14 experiments. So you can get neural stem cells that you can take out of the adult CNS. There's stem cells that cycle in vivo, but you can also pull them out, even from regions where there is no apparent neurogenesis, and you can coax these resident neural stem cells, even though they're not active in that region of the brain, you can get them to expand and differentiate into neurons and glia in culture, right? So there's been a lot of progress. We know there's stem cells. This is only in the last 20 years. Now we know they used to think there was no more brain growth, but we know there's stem cells in the brain, and also we can get cells to grow outside. But there are no observed neural stem cells in the peripheral nervous system, right? So the peripheral nervous system is derived entirely, as far as we have known up to now, from these neural crest cells, right, which migrate out of the neural tube. And as these neural crest stem cells are found exclusively, or not exclusively, but are found outside of the CNS, and that's what accounts for neurogenesis outside the central nervous system in the peripheral nervous system. And even these neural crest stem cells, they have a limited capacity for self-renewal, and they're very different transcriptomically and functionally, and differentiation potential from bona fide neural stem cells, right? And the dogma was that there are only neural stem cells in the central nervous system, and the neural crest stem cells are what account for peripheral nervous system, right? So that was what we thought, but now we know that that's not true. And I'm going to cut to the chase, because the way they found this is, I think, is a cooler element of the story. So this is a story from Hans Schollers at the Max Planck, along with Dong Han and Pentao Liu, who are at the University of Hong Kong. They're all co-corresponding. And I think that it's probably because maybe one of the first author, Dong Han, must have originated in the Scholler lab. This story was submitted 5 years ago in 2020. And the initial observation was made 10 years ago, more than 10 years ago. And I'm going to come to that. But the punchline here is that they found that, yeah, in fact, there are peripheral neural stem cells. So neural stem cells that exist outside the central nervous system. They show that they're different from neural crest stem cells. They have a lot of these bona fide neural stem cells. I said they're different from neural crest stem cells, I hope. They're similar to bona fide neural stem cells in terms of their marker expression, the morphology. They self-renew and differentiate like neural stem cells. Their transcriptional profile, epigenome, is very similar to these control neural stem cells. They show by this is all in mouse, they show by lineage tracing that they are a SOX1 positive cell that originates from neuroepithelial cells in development. So they do have their origin from the same part, but then they move out into the peripheral system and can be reactivated. They differentiate into mature neurons and glial cells, even in vivo. Like, even though we didn't know it, these neural stem cells, they do become mature neurons and glia outside of the central nervous system. We just hadn't observed it up until this point. Now, here's what I think is so cool. The way they found this is because they were trying to reproduce the STAP. Remember the STAP, stimulus something acquisition of pluripotency, that led to the unfortunate scandal and suicide of Yoshiki Sasai. It was retracted way back in 2014. Well, the Scholler lab and other members on this paper, presumably, they were trying to recapitulate STAP, and they labored at it for however long. And then the paper was retracted. They were like, oh, okay, I guess that didn't work. No wonder we couldn't get it to work. But in one condition that they used, they found there were these weird neural stem cells that were induced by low pH. They did the STAP protocol, low pH. So it turns out, I think, in some tragic irony, that STAP was not a total fraud. STAP actually was able to elicit this important biological result which not for nothing provides some insight, but also like to get neural stem cells from the peripheral system, not having to go into the brain. Yeah, like that's a pretty big deal as we move toward, uh, cell therapies in the neural space, not having to go from a pluripotent progenitor. Like that's a big deal. So this is a story with a lot of impact. Of course, it's in mouse, and we'll have to see. But I love the narrative here. I just pity whoever submitted this 5 years ago and has been slogging along, and a story that's been fully a decade in the making.
Yeah, this is one of the more bizarre stories that I think we've covered here on the Roundup in a long time. In part because of the history, like what you alluded to, the origins of this observation are dark, right? Because they are coming from STAP, which, as we know, was a terrible scandal in the pluripotent stem cell field and really a black eye for that particular sub area of research, and led to some terrible consequences. But something potentially good came out of those initial STAP studies, and that's this observation of NSCs. And I'm not a neuroscientist. You're not a neuroscientist. Maybe this is something that we'll have to talk to about with Sergio, for example, or Clive Svensson, or whoever, many neuroscientists who are in this particular area, about the reproducibility of some of these observations. And like you said, we have to extend this to, of course, human as well. But this is a dramatic observation, very big deal, because the assumption was that these NSCs are found truly in the CNS, in the central nervous system, right? But they're doing things elsewhere in peripheral populations and turning into stuff that they are supposed to, not in their traditionally accepted niche. So this is more a reflection on, again, the plasticity of the human body, the importance of the niche, the developmental potential of these cells that are outside of, quote, where they're expected to be. But I do think there's going to be a lot of eyes looking at this paper very critically, don't you think?
Oh man, absolutely. You make a very good point. A lot of good points there. The implications and plasticity. I think that's the major takeaway for me in terms of like conceptual insight. Is like every cell, you know, who knows what the potential of a cell that is latent, in whatever niche. But yeah, the scrutiny moving forward is going to be high. But it occurs to me that the reason why this paper was in review for 5 years, is because they really probably put the feet to the fire. In fact, it must have been an exercise in courage to even make a claim with STAP. I mean, it takes a lot of courage to get out there with something that I guarantee you a lot of editors, were probably like not going to touch that with 100-foot pole. We got to talk to Stelios about this one.
Yeah, this is Nature Cell Bio, so I'm actually very curious about what Stelios had to go through for this particular paper, what the authors had to go through, but probably not something we can discuss online. We have to discuss that maybe at the bar at ICCR, for example. Again, June 11th to 14th. Check it out. Moving on to the last paper, the roundup for today. This is not a neuro paper. I thought we were going to go 4 for 4, but this is a kidney paper, so sorry about that. This is a paper in Science Translational Medicine, and you guys know I love this particular journal, titled Human iPSC-Derived Nephron Progenitor Cells Treat Acute Kidney Injury and Chronic Kidney Injury in Mouse Models. Importantly, right there, mouse models. So there's a great need for this particular field of work. Of course, there's not a whole lot of effective treatments for chronic kidney disease, and also same kind of deal for acute kidney injury. I mean, people have to be on dialysis and all this kind of stuff. It's really debilitating. So the thought is you might be able to use pluripotent stem cells to be a therapeutic option for both CKD, chronic kidney disease, and AKI, acute kidney injury. So folks have made iPSC-derived nephron progenitor cells. I think we've actually talked about some of these studies on the podcast. And the potential for these NPCs, these are not neural progenitor cells, these are nephron progenitor cells. The potential of those cells on AKI, acute kidney injury, have been reported previously in mice, but certainly not confirmed clinically. And there's also not whole bunch of reports, a few, and examining the therapeutic potential of these iPS-derived nephron progenitor cells on chronic kidney disease, right? So the two sides of the coin, acute and chronic, and when it comes to the kidney injury. One of the big concerns here is the expansion of the iPSC-derived nephron progenitor cells is apparently pretty tough to scale these things up, and you need a lot for cell therapies, of course. And expansion cultures remain to be developed, effective expansion cultures. And here, this is a tech development story, more of a tool development approach, more than anything, because they created a culture medium for these cells that can enable more than 100-fold proliferation of their of the NPCs from multiple IPS lines. And get this, just two passages. That's pretty amazing. So I'm not going to dive into the actual recipe, the formula here, but just know if you're working in this area and you have problems with expanding IPS derived nephron progenitor cells, you might want to check this out. Or you probably already are checking this out. So they named their medium CFY, or by their conditioned medium alone, attenuated the kidney injury, so it improves survival after expanding the NPCs, right? Improves survival in the cisplatin-induced AKI mice, acute chronic injury mice, kidney injury. And they also observed that these NPCs that were expanded in this media prevented the functional decline of the kidney, interstitial fibrosis, senescence, and acid induced CKD mice. And they also took a deeper mechanistic dive into it, as you might expect, of how these NPCs are actually doing their job in ameliorating the kidney injury. They found that c-Met is a specific cell surface marker for these NPCs and confirmed that purified c-Met c-Met positive NPCs had a therapeutic effect on both aspects of kidney injury, the acute side of things and the chronic kidney injury. So it's a tool development story, like I said. And finally, they found that the NPCs exerted their therapeutic effects in chronic kidney disease and acute kidney injury by secreting something that you know and love, Dalen. That's VEGF-A, vascular endothelial growth factor A. There you go. So it's like I said, tech development. You're using this custom media to expand these iPS-derived neuroprogenitor, not neuroprogenitor, nephron progenitor cells. Caught myself there. Expand these cells for potentially for cell therapy, in vitro modeling, maybe because it's so hard to expand these things normally. And it might open up new avenues for treating kidney disease. That's ultimately the hope here.
Yeah, I see whenever I see, and it's more and more, you see a stem cell, pluripotent stem cell story in Science Translational Medicine, you get excited, right? Because I mean, it's not like Nature Medicine where it's like in people, the story has got to be something that's in people already. It means that it's like it's been optimized. The story is going to be like, okay, here's why we're ready for prime time. So am looking forward to see how they spin this into a large animal trial or something like that. I see Juan Carlos on there from Altos Labs. So as you were saying, this is like the tune-up where they're optimizing their systems and getting ready. It's almost like an industry paper. But here, I think they really deliver on the science as well. And as you said, also addressing both acute and chronic, is showing that they're really casting a wide net. And while, media stories are, the little nitty-gritty type details, I mean, like VEGF, only way I would have been maybe less surprised if you'd said Wnt. But like, I get it, maybe not the sexiest story or the most bizarre coming from our, STAP offshoot there. But these are the stories, right? These are the stories that are the fabric of our translational adventure. And this is the prerequisite to seeing this be applied in larger animals and ultimately humans. So I love every time. It's such a great time to be reading stem cell stories because it's every day you see a story like this. Okay, looking forward to the future without kidney disease. That's another thing that they fixed for us. So a really great story in a great journal.
Absolutely. Hopefully, folks can be not tied to the dialysis machine in the near future. We'll see how things go. I mean, and the other thing to highlight here, this is anytime you're talking about the cutting edge of iPSCs and their application towards clinical therapy. And I think there's a few institutes in the world that are, leading the charge here. And this is a paper coming from CIRA. I had to highlight that, of course. CIRA, very closely tied to the guy himself, the man himself, Dr. Shinya Yamanaka, who first derived iPSCs and described them. This is a very closely tied institute to him. They're doing incredible stuff over there in Kyoto. And I do know next year, shameless plug, ISSCR is having an amazing event at CIRA and at Kyoto, celebrating the 20-year anniversary of the derivation of pluripotent stem cells. So hope to see you there. I mean, I will for sure be there.
Well, we're about to get to our interview with another man himself, Professor Hank Greely, who I love. I love a chat about the bioethics. It's probably going to go long because there's so much to talk about. But before we get there, I got a quick message from STEMCELL Technologies. Do you require large numbers of human pluripotent stem cells for your research applications? Learn how to expand and scale up HPSCs in 3D suspension culture with this free on-demand course. To learn more and register, visit stemcell.com/3DPSC course. All right, you guys, with us on the show today, we have, I would call him a special guest, specifically because we're going to talk about a bit more far-ranging subject matter today on the show, all related to stem cells, of course, but we get some real big picture conversation, hopefully, here, because we have with us Professor Hank Greely, who is the Deane F. and Kate Edelman Johnson Professor of Law, also professor by courtesy of genetics at Stanford, also the director of Stanford Center for Law and the Biosciences. Professor Greely's current interests in human genetics include prenatal genetic diagnosis, the impact of widespread clinical whole genome sequencing, and the ethical, legal, and social issues associated with genomic biobanks. In stem cell research, he focuses on legal challenges and the complex questions surrounding human, non-human chimeras. His work on human research protections, biological enhancement, and the future of reproduction intersects with and draws upon all three of these areas. He's really thought about a lot of the subjects that we kind of, incorporate into our worldview. He thought deeply about these in terms of the ethical impact of these. Professor Greely, thank you for joining us on the show today.
My pleasure. I like to talk.
Thanks so much for being here. It's great to catch up with you after a few years at Stanford. You've been at Stanford way longer than I have. I was only there for 4 years, but you saw the dramatic evolution of the stem cell field and biomedical research as a whole. I mean, I remember when I was a grad student in the stem cell PhD program, you provided some really cool and thought provoking lectures to our cohort, especially for some of the junior students. So I really appreciate that. I mean, like Dalen said, you've been at the forefront of analyzing the bioethics of pretty much every single cutting edge technology that's emerged over the years, ranging from genome editing, iPSCs, animal chimeras. But before we dive deeper into each of those, could you actually start off by telling us your path to becoming a bioethicist at Stanford? And, for our trainees considering a career in the field, what it's like to be a stem cell bioethicist these days, like a day in the life, so to speak?
Well, it's hard for me to give you a day in the life of a stem cell biologist since I'm not a stem cell biologist and not even a biologist at all. My last bio class was in 10th grade when we barely knew how to spell DNA, and they were actually just beginning to figure out which codons meant which amino acids back in 1967, I think, 67, 68. My path to what I do is like most people's paths in life, some combination of interests and random chance. Uh, I always, as a kid, was really interested in science, but I was also really interested in sort of government and policy. I was an undergrad at Stanford. Fall of 1970 was the first of my now 44 years in one capacity or another at Stanford University. And I thought I would either major in science or major in something policy related. I ended up majoring in political science. So I did major in science, but political science. Ironically, most of the science I actually work on turns out to be very political. So it's a little bit appropriate. And in part because I like science, but there wasn't one particular science that grabbed me. Plus, my native math ability deserted me without warning in the second midterm of the third quarter of the FAST Calculus and Analytic Geometry course, and I overreacted. But I continued to I went to law school, didn't do anything with science in law school, but continued to read about it and like it. After law school, I worked for a while in the federal government, both in the Carter administration, both for federal judges and then in the Carter administration. And then on January 20th of 1981, I don't usually remember the dates when I leave jobs, but that was Inauguration Day. I decided to quit before I got fired the next day by the incoming Reagan administration and went back to California to practice law. Two important things happened there. I learned that I really didn't love practicing law, and I learned that I really did love the sister that one of my fellow associates introduced me to on a blind date. She was a resident at LA County, first a resident in internal medicine and then a pulmonary fellow. We got married. She hated my job. I didn't love my job. I applied for teaching jobs. I got incredibly lucky and got a chance to teach at Stanford, where they hired me to teach oil and gas law. Because Stanford had had the country's two leading oil and gas scholars, and they'd both retired. And I had worked at the Department of Energy, which had nothing to do with traditional oil and gas law, but who was I to correct the Stanford faculty on that? However, having married a doctor, I started teaching health law. In 1991, about the time I got tenure, I decided I turned the oil and gas law into energy law, but I decided I couldn't keep up with both energy law and health law. I couldn't keep up with either one of them on its own, but the two combined were impossible squared. So I picked health law on the theory that in energy law, the big issue would be climate change, and there was no way we would have the political will to do anything important about it for the likely 30 years of my remaining career. Health law, I said, we're going to get a Democratic president in 92. We're going to get universal health coverage. That's going to be really exciting. So I think I was kind of right and kind of wrong on both of those things, but I picked health. The Clinton health plan crashed and burned comprehensively. But luckily for me, the Human Genome Project came along. University put on a 3-day symposium on the then new Human Genome Project. I was put on the planning committee because the scientist wanted somebody from the law school. And I was probably the only person in the law school who could spell deoxyribonucleic, maybe even spell DNA. Had a wonderful time on that committee with Paul Berg and David Botstein and Lucy Shapiro. And met a bunch of geneticists who started asking me questions. That was 30 some years ago, 33 years ago, and I haven't looked back.
Wow. Talk about a random walk, but
well, but not entirely a random walk. You know, there was that predisposition to science, which I think came from loving science fiction as a kid.
Yeah, oh, for sure. I think all of us have that central, core element in us that keeps us in it or keeps us open. But it's just interesting to me how your walk, so to speak, kind of reflected a lot of the environment. And we talk about stem cells in the niche. We love to pull out the metaphors, but we are all in our way kind of this multipotent stem cell just reacting to the niche, right? And that's a really fine illustration of it. But I can already tell this is going to be a fine conversation. I'm enjoying it already, but I've been looking forward to this one because I've really enjoyed the conversations we've had in the past with ethicists, that focus in some tangential or direct way on stem cell research in biotech, whether it's talking about bipaternal offspring with Insoo Hyun last year. That was episode 243, if anyone wants to have a listen. Are mulling over the legal implications of stem cell therapies, specifically with Ubako Ogbogu, which is way back, episode 148, for all of you looking out. Whatever it is, the chats are a highlight for me because they speak to the kind of broader implications of the work, while also reflecting, as I kind of alluded to in your walk there, while reflecting the pace of technological advance. Like the questions we think about and talk about are the questions of the moment. But maybe we don't talk about enough the progress of bioethics itself, as a field. We all know the iconic, black mark in the history of research, Tuskegee. We've come a long way there from an institutional regulatory standpoint. And the guidelines, I would say, continue to be revised. Look at the ISSCR. continually coming out with these revised guidelines, kind of keeping pace with our ability to model human development. But I want to ask you, as someone who has a real big picture view, as your walk, again, going back to your walk, kind of illustrated, you've seen over the decades kind of how bioethics has evolved. What's really at stake, right? Why do we need this robust framework for a continuous and vigorous consideration of the ethics, even now with the IRBs and everything that we have in place?
Great question. So first, I'd say, like every word, ethics means many different things to people. I take a very broad view of ethics. We talk often in my world about ethical, legal, and social issues or implications, ELSI. But I view ethics as not just is it right or is it wrong? And bioethicists not as sort of judges in black robes who say, you go to hell, you go to heaven, but more people who help science and the society understand what the implications are and how it fits in to society. Ultimately, science is going to be part of its society. It's going to be, in the long run, controlled by its society. It needs to stay in touch with the society. And one of the, so what a important thing about bioethics, obviously, is to try to do the right thing. Scientists are people. Scientists are moral actors. Scientists want to do good. They don't want to do evil, with the occasional movie exception. So you want to do the right thing, but you want to think about in some of these areas, what's right isn't immediately obvious. And so thinking through it harder and seeing the different sides is one of the things bioethicists can help science do. But the other thing is, at least for me, keep the public and science in touch with each other, make sure that the public understands why scientists are doing what they're doing, what the implications are, and to say make sure the public understands. That's way too ambitious. But do your best to, as you're doing with this podcast, to some extent, to make sure that scientists and the public kind of understand what the issues, know something about what the issues are. And so one of the things I think that we do a lot, and this varies from the person doing bioethics or the institution doing bioethics, but at Stanford, I think one of the things we do really well is to try to help science to see what the ethical issues are, how they can perhaps modify their work in ways to avoid some of the ethical issues, and how they can present their work publicly in a way that keeps them from blowing up in their face. I mean, the example I always remember is Dolly the sheep, which was in a way my first introduction to stem cells, even though Dolly wasn't really a stem cell story, except kind of she was. But Dolly led to lots of discussion about cloning for about the same time that Jamie Thompson figured out how to keep human embryonic stem cells. When Dolly hit, she was a shock. Nobody expected her. There were hysterical headlines all over the press, all over the world. And people immediately went from one sheep to armies of cloned warrior slaves. Countries and states, including California, all over the world, passed laws banning human cloning. And some of those laws were really stupid, but none of them was done, but they were all done in that sort of semi-hysterical mood of don't just stand there, do something. That's a bad thing. Scientists and science needs to be in a conversation with the public so that both the scientists can see what the public reaction may be, and the public can know what the science is planning and where it might go. And most importantly, I think, why scientists are doing it. The Frankenstein myth or the Faust myth, the idea that there's some evil, reckless genius there doing things just because it's fun, is almost never true in science. It's, there are bits of it here and there. But you got to get grants, you got to get funding, you got to get tenure, you got to get all these things that constrain you. You've got to get approval from an institutional review board if you're using humans, or an IACUC if you're using animals. So helping the public understand, I'm not doing this just because I think it's cool, but because by learning about how these stem cells in mice become different organs, we can learn something that might teach us how to repair or build new human organs to treat disease and relieve human suffering.
Yeah, I think it's something that, of course, you've been doing for a long time in terms of properly communicating the science to both fellow scientists and also to the general public and relaying the importance of these emerging technologies. It's like you mentioned, it's something that we hope to do here on the show as well. We're not, fully public facing. We cater to mostly stem cell biologists here, but it's still a very relevant topic of conversation because there's so many incredible new technologies just within this particular field that have emerged in the last 10 years that are extremely worthy of dissecting their moral and ethical implications. And one of those technologies that we talk a lot about is, of course, organoids. And one of the hottest topics in stem cell biology. I'm a cardiac biologist. I make cardiac organoids. And we've talked to so many folks in neuroscience who make, these cortical or brain organoids from pluripotent stem cells. We've had Sergio Pasca, also from Stanford, here on the show many times. We talk about the caveats about these things, and as stem cell biologists, we're aware of them. They're nowhere near as the complexity of the real human brain. But as these things become more complex, like Sergio Pasca is working on these assembloid structures and so on, and combined with other organoids, what concerns about their use or around their use do you think are particularly important to focus on? I mean, again, they're nowhere close to the real deal, but I think it's definitely worthy of discussion.
Right. So I've worked a lot on human neural organoids. I work very closely with Sergio. I think and I was on the National Academy of Sciences committee that put out a report on organoids in 2021, about the same time that the ISSCR came out with its revised set of guidelines. I think the biggest concerns are in the future. Right now, nobody thinks that an organoid could feel pain or could, wake up one morning and say, gosh, where am I? I can't see anything. But in part because we don't really know what in our brains causes us to feel pain or to be conscious of ourselves. It's pretty hard to say, when is that threshold going to be crossed? Right now, some of the human neural organoids are 1 million, 2 million, 3 million neurons. A bumblebee has 1 million neurons. A bumblebee engages in some pretty complicated behavior. And I think pulling the wings off a bumblebee would probably hurt it. Now, an organoid doesn't have a brain like a bumblebee's, let alone a brain like a person's. But as they get bigger, either because we learn how to vascularize these, so they're no longer restricted to small hollow spheres, or as Sergio is doing, we string them together. I think of them as like pearls on a string, on a necklace, and connect them. If you connect 23 million neuron organoids, you've got 60 million neurons, which is about the same number of neurons in a mouse brain. Now, they're not a mouse brain, but what are they? What can they do? Exploring that, worrying about that, thinking about how could we even study that is really interesting and important. One of my friends, actually a sociologist of religion originally at UC San Diego, John Evans, wrote a book about public reactions to organoids. And this sort of floored all of us coming from the science and bioethics world. We thought that people would be mainly worried, as we were, about consciousness and sentience and pain and so on. He found that the biggest fear a lot of people had about organoids was those cells started out from somebody. So if we made a human neural organoid from Arun Sharma, would it remember Arun's past? Would it have Arun's personality? To what extent would it be Arun? Now, the answer is almost certainly not at all, other than DNA. But people worry about those sorts of things. And so I think it's both a case of explaining what it is, what can be done, and worrying about how to test for the things that people are concerned about. But I don't want to say it's just about explaining. The relationship between science and the public can't just be scientists lecture and the public nod, not nod off, but nod understandingly. It's got to be an interaction. So scientists also have to listen to the public. That doesn't mean that they let the public decide entirely what they do. Although I would note, in the long run, the public, through Congress and through NIH and others, and the president, does ultimately decide what most scientists can and can't do. But it does mean that you listen to the concerns, discuss the concerns, react to the concerns, sometimes change your research in some ways, not in ways that jeopardize the quality of it, but that make people more comfortable with it.
Yeah, that I think a lot of scientists, so Arun and I talk oftentimes how we're kind of in our silos, and it's kind
Yeah.
of to our own detriment nowadays, it feels like. And as you talk about that interface, the exchange with the public, I think that's maybe what we don't think about enough in terms of the ethics, is like really understanding what the ethical implications of our work are by way of the public, by engaging with them. And that I was very, I would say, naive to that, almost completely before. Just in the ISSCR this past summer, I attended this policy ethics session. Um, this is at the meeting in Hamburg. And it was this panel on the governance of embryo model research, which, I was in it for the science, but it really opened my eyes to how important these questions are, and how we have to consider them now. And I'm remembering probably poorly and synthesizing a bit loosely here, but one of the panelists had gamed out a complete framework for governance of ectogenesis, right? We're talking about the complete growth ex vivo of a human, from zygote all the way. And I think a lot of people are like eye rolling or maybe laughing, but the panelist was explicit, like, yeah, it may seem like a joke, but the rationale for doing this is, regardless of whether or not this thing is practical, you have to set up the framework for thinking about it long before you can actually do it. And that was such a revelation to me. And it kind of, I think, reflects what you were just saying about how you have to think about these things. Maybe it's not now, but in how long is it going to take before you have a threshold of neurons for something that you have to think about? This is a real possibility.
Right.
So what do you do? Like, do you find yourself, like, just out of curiosity, for me and probably for our listeners, do you just game out all these far-flung possibilities? Or is there enough now with the current menu of like possibility and practicality that like there's a lot to think about already? Or are you like thinking of these really kind of maybe sci-fi scenarios?
Yes. I mean, A or B, the answer is yes. A little bit of both, although I try to stick to things that I think are plausible within the next 30 years. Plausible, not certain, not even necessarily probable, but plausible. And I'm curious who the panelist was who was talking about ectogenesis, because I'm currently writing about ectogenesis. And I have recently decided there's a pathway that could lead to clinical trials for complete human ectogenesis within a decade.
Holy.
When in the end of sex, I put that 40 to 50 years out. So being open to plausible paths forward and then thinking them through, we come up with frameworks and guidelines. They're never going to be the final ones. Niels Bohr supposedly said it's always hard to predict things, especially the future. Turns out it was probably a Danish writer named Storm Peterson. And of course, it's wrong. It's easy to predict the future. It's just hard to be right about your predictions. When we set up these things in advance, they'll never really be perfect or be right, but it gives us a place to start. It gives us a way to be, it gives people notice that this is an issue to think about. So there are problems with going too early. There are also problems with going too late. Actually, the issue about embryo research is, I think, a nice example. In 1979, when an NIH committee recommended a 14-day rule, and then in 1984, much more influentially, when the Warnock Commission in the UK suggested strongly a 14-day rule for research with 14 days of non-frozen, 14 days of development as a endpoint for research with human embryos. That was pretty easy for science to agree to, because we could barely keep embryos ex vivo alive for 3 or 4 days. So it's easy to give up things that you can't do. But by 2016, people were beginning to be able to keep human embryos alive ex vivo for 13 days or 14 days. And we had to sort of confront that kind of question. So flexibility is really important, thinking about things early, but not thinking you're going to come up with the final solution. It's an early step in a process. And I think, I applaud ISSCR. I think the guidelines this is the third set. They've been revised twice, I think, in 2016 and 2021. And I hope they'll be revised again soon because things move fast and things change. The science changes, but also the society around the science changes. And the right policy, good advice is a product of both the science and the society.
Yeah, absolutely. Very, well said. I mean, you also mentioned the book that you wrote a few years ago, The End of Sex and the Future of Human Reproduction, where
Yeah.
you discuss some of the potential of these, new reproductive technologies to fundamentally change the human species, kind of like what we're talking about. I mean, something like IVF, of course, has been commonplace for decades now. And well, recently though, we've
I,
had
I,
a,
I was adult when the first IVF baby was born, and
oh,
I am old but not ancient. So yes, from your perspective, it's been around forever.
yeah.
But it's every IVF baby ever born could drop dead on her 47th birthday
Hmm.
Hmm.
because the oldest baby, Louise Brown, is only 46. We
Hmm.
don't know. No reason to think she'll drop dead at 47, but
Right.
we don't
Fingers
know.
crossed.
Yeah, that's a good point. But it's more than just IVF these days. You have other technologies intersecting with IVF now, right? As for example, genome editing, right? And unfortunately, we had that first birth of the first genome edited babies in China, which was quite controversial when it happened a few years ago. And I mean, that's something we could talk about, of course, forever for its own podcast. But in general, how do you think advancements in genetic engineering and stem cell research might reshape the future of human reproduction, like what you alluded to in your book, and what other dilemmas might emerge as these technologies become even more mainstream, right?
So that book was about what's now become acronymed as IVG, in vitro gametogenesis, making eggs and sperm from induced pluripotent stem cells taken usually from fibroblasts from the skin. And it's a really fascinating topic, and I think it's going to happen. We aren't there yet, but any morning I could wake up and find that the proof of principle paper has been published or at least pre-printed saying we have made what looks like a fully functional mature sperm or egg from human iPSCs. It's been done with mice. We aren't mice, but we aren't not mice entirely. There doesn't seem to be any obvious reason why it shouldn't work in humans, and people are getting closer and closer. Like most final cells, it's the end result of a long cascade through various stages. The stem cells become different, more specialized stem cells. And I think with the oocytes, we're down to about 2 steps away from mature oocytes. And with sperm, we're 2 or 3 steps away. And, 10 years ago, we were 10 steps away. So I think that in itself would have major implications, mainly for people who desperately want to have kids that are genetically half theirs, but who don't have eggs or sperm, or don't have eggs and sperm that work or that work anymore. That's a lot of people. It also, though, if you could make eggs and sperm from cell lines, egg shortages are a thing of the past. And I'm not talking about buying eggs at the grocery store where the shortages are the thing of the present and the future. But human eggs for research and other purposes, if you do an egg harvest from a healthy young woman, you on average get between 0 and 25 eggs, usually roughly 10 to 15. If you're making eggs from a cell line, how many eggs do you get? How many eggs do you want? Hundreds, thousands, potentially millions. So one of the things I did in that book was combine it with pre-implantation genetic testing and say, in the future, people will make 100 embryo, will make a bunch of eggs. They'll probably get sperm the old-fashioned way because that's cheap and not dangerous and not uncomfortable for the most part. But they'll make 100 embryos. They'll get whole genome sequencing on each of them, and they'll pick the ones they want. And I think that side, genetic selection of embryos, is already here. It's been here for 30 years. It's going to get even bigger. It, like every new technology, has its problems. There are a couple of companies that are using probabilistic risk scores for embryo selection. I am very unhappy about that because I think PRSs are not well enough attested, are not well enough proven to be used for something like that. But it's happening in an unregulated environment in the United States. It's regulated elsewhere, but not here. So, on the other hand, I think parents choosing to avoid Tay-Sachs disease or sickle cell or cystic fibrosis or a variety of other nasty diseases is clearly a good thing. And I'm not all that worried about or upset if parents decide to use as a tiebreaker in choosing embryos, hair color, or eye color. It's not a big deal. I am less worried than many people because I think enhancing behavioral traits, there's the kind of enhancement we care most about, I think. Enhancing brains, enhancing intellect, which I think academics particularly prize, but sports ability is in your brain to a large extent. Music ability, math ability, art. If we could enhance that, then you get into the science fiction stories about designer babies, super babies. And in theory, if you have 1,000 embryos to choose from, you can pick the ones that look best for sports or for art. Except that brains are really complicated. Brains make genetics look simple by comparison. And I don't think we're ever going to be able to do, well, certainly not during my lifetime, and Arun, I think not even during your lifetime, are we going to be able to do a very good job of predicting cognitive or behavioral traits strictly from DNA. A little bit at the edges. So in the book, I say in 20 years, you'll be able to say, well, this embryo has a 13% chance of being above the 90th percentile on its SAT scores, which of course means it has an 87% chance of not being in that top decile. Is that important? I don't think so. So I don't worry that much about super babies because I think brains are too hard, at least for the rest of this century, which is now a quarter of the way through. But I think embryo selection is going to be a big deal. My second book, CRISPR People, about Hu Jiankui and the 3 babies, whose health, by the way, we know nothing about. They are now almost 7 years old, well, 6.5 years old. We don't even know whether they're alive. He says that they're healthy, but He is a known liar. China has been extremely closed-mouthed about those, and we don't know what their genotypes are. We don't know what their health status is. We don't know anything about them, which is deeply frustrating. I don't think the genetic editing of embryos is going to be that big a deal, in part because most of what you want to do, you can do through embryo selection, certainly with most monogenic conditions. Much of it can now be, or in the future, be handled through post-birth or even in utero therapy. So as gene therapy or as alternatives to gene therapy get better at treating genetic disease, you don't have to worry about editing embryos as much. But if you have multigenic traits and you need to have the right combination of 40 different alleles, selection gets really hard, especially if the parents don't have the relevant alleles. Editing's your answer there, but it's only an answer if you actually know enough to go after complex multigenic traits with confidence, which I don't think we'll be able to do for a long time, if ever.
Yeah, I mean, look, the assisted reproduction, I have a deeper connection, both because, I mean, this isn't so deep for me, but it is true. I was, among the last babies to be born in the pre-IVF era, as I was born just a month or so before Louise Brown herself. But I also work in an IVF clinic, and I do note that, it's, I would say, the vanguard of medical innovation, reproductive tech. But it's also, I would say, it skirts controversy a lot. And it's kind of strange in terms of application and translation of technology, because by the time kind of the ethical implications of some things sink in, the tech's already out of the gate and there's babies walking around, like ICSI, for example, right? So it's kind of this arena for beta testing these societal changes
With
that
our most
are real.
precious things, with babies.
Yes, with
Right.
with not only our babies, our germline, right? So yeah,
Screw the germline. It's the babies that are important. And,
they're
and
very
I,
they're
I, I
very
mean
cute.
that. I mean, I actually mean that. I think there are people who hear the human germline genome and think about like the Ark of the Covenant from
Yeah.
Raiders of the Lost Ark.
Yes.
Amazing, immense power, holy thing. There is no human germline genome. There are 8 billion human germline genomes, and they change every generation because we don't inherit exactly what our parents inherited from their parents
But
because of somatic mutations during their lifetimes in their germ cells. So, yeah, it's a big deal, but it's not a holy thing. The only, there are people who think it's like the Ark of the Covenant. It's immensely powerful, and we need to keep it from away from the Nazis. Well, I'm in favor of keeping it away from the Nazis, but
I agree with
I
that
don't
too.
think it's going to be. I don't think germline editing in humans is going to be that important. But your bigger point about risk in IVF,
Well,
I
my
mean,
bigger point
is interesting.
is
Is a good one.
it's less about risk because I recognize the risk, and I, like you, I think that it's a pragmatic solution to a real problem. And as you allude to there's a lot of kind of germline editing, you might argue, just extant in the world today, given all the environmental exposure. So the
Right.
idea that we have some kind of precious germline that is, going back however long, I agree with you that that's overstated, but I think it's fair, and you would agree that this is something that we need to pay attention to. But my greater point is more about this beta testing idea, and
Yeah.
really your idea as someone who really understands not just the ethics, but how do you communicate ideas, and how do you communicate them over time, and how does the appreciation of not just the public but the stakeholders, how does it evolve over time? I take, for example, the I would call it kind of the marketing of sex selection. When I joined my clinic, it was very distasteful. People would talk about sex like a taboo. Oh, I don't. Some of the attendings, I don't practice that. Everybody knows the guy who does it, and they refer
Right.
the patients. But now, uh, no longer called sex selection, called family balancing, and family balancing is commonplace.
Yeah.
Everybody,
You,
it's
s- you,
universal.
s- you see it in changes in the guidelines from the American Society for Reproductive Medicine, which is kind of the ISSCR of assisted reproductive technologies. They were against it, then they were in favor of it in certain limited circumstances, and now it's more an open world. But even more fundamentally, there were serious people who opposed IVF altogether, and not just old men in the Vatican. There was worry that IVF babies would be discriminated against. Parents were very reluctant to let people know that their kids were born as a result of IVF. If there are 10 million people in this world who owe their existence to the fact that Louise Brown was born healthy.
Mm.
If she had been born with some serious problems, IVF would have been set back enormously. But she wasn't born with horns and a tail.
But I mean, you joke, I know you're serious, but that's your point being that it really was key that it worked out. And there have been
Yeah.
kind of fumbles, right, with gene therapy that have illustrated how it can get screwed up. So I
Well,
think the question to you is someone who's seen kind of the missteps and maybe I call it lucky or call it careful, whatever it was with Louise Brown. Is there for
little
our
both.
kind of younger scientists out there who are kind of moving the field, do you have to do you have to think about the marketing of the therapy in advance? Do you have to brand it as family balancing before you actually get it into the public sphere?
So I think you need to think about the marketing, even if you don't intend to have anything to do with the marketing. You need to think about how people driven by profit are going to try to make money off of your discovery, and whether you think that's a good thing or a bad thing, and how it might be mitigated if it's a bad thing. Personally, I am in favor of more regulatory approach. I think IVF, we got incredibly lucky with IVF that it's turned out to have, apart from multiple births, really very limited, at least gross safety issues. There are some subtle questions that I think are still out there about the safety long-term of IVF, but at a broad level, it seems to be safe. But Steptoe and Edwards didn't really know that. They hadn't done that much research with non-human models and so on before they started trying it in humans. We got lucky. I think it should have gone through a phase 1, phase 2, phase 3, preclinical phase 1, phase 2, phase 3 set of FDA rules, but FDA didn't want anything to do with it and still kind of doesn't, even if the FDA is going to continue to exist, which shockingly is kind of an open question, whether it exists de jure, under law, or whether it exists de facto in reality. The de facto part is getting iffier and iffier with every passing day. So I think individual scientists should think about what could be done with their work. Do they think it would be good or bad? And how can they influence that, both in how they write things, how they brand things, what their associations do, things like the ASRM and the ISSCR. Those do useful things. National Academy studies do useful things. So it doesn't have to be hard law. It can be science-based or scientist-informed or crafted guidelines, but those things are important. They're important in providing some early boundaries, but also they're important in starting the communication and the conversation with the public. That's going to be crucial. And I do think that IVG, in vitro gametogenesis, it would be a real shame if that starts getting used without adequate proof of safety. Making disabled babies, that's a really bad thing. You don't want to do that. Now, you can never prove complete safety. The complete safety is always impossible. But say, as safe as IVF, that would be a nice standard to look at. But of course, we don't really know exactly how safe IVF is because we haven't done 1,000-person clinical trials. So, yes, we need to think about the future. There is something called the Colin-Ridge dilemma that I think is little known that I want to mention here. I forget what Colin-Ridge's first name was an engineer, and I think back in the 80s, he pointed out there's a real problem. The best time to regulate a new technology is at the very beginning, before it's developed vested interests and companies and patents and journalists devoted to it, because then you've got the least political pushback for regulation. But the worst time to regulate a new technology is at the beginning because you don't know squat about it. You don't really know what's going to happen. By the time you really know what's going to happen, though, as is the case with IVF, you've got hundreds of clinics and tens of hundreds of thousands and millions of parents. And it gets really hard to regulate. So that's the Colin-Ridge dilemma. The time when it's easiest to regulate, you know the least. The time when you know enough, it gets really hard to regulate because of the sort of, you let it already out into the world. And once it's in the world, it becomes much harder to control.
That's David Colin-Ridge from his 1980 book, The Social Control of Technology. I'm going to have a look at that.
Well, I got the decade right. And the last name.
There you go. I mean, one thing you alluded to, no matter what the technology, emerging technology is, dialogue is critical. I mean, we have to do our best to try to get ahead of some of these issues. Whether we can or not is another question, but we got to have the conversation. And this has been an incredibly stimulating conversation for all of us here, for both of us, but I'm sure for all of our listeners as well, especially the junior trainees, folks who were in my shoes way back in the day, who were, like I said, I was listening to you when I was a first-year grad student, and that was really formative for helping me think about some of these technologies, because there's just so much to think about. And even in the past 10 years, there's been an explosion in these incredible game-changing technologies in stem cell biology, whether it's sperm and egg derived from iPS potentially, or genome editing, or organoids. There's always going to be something to talk about. So we're so glad that you're here chatting with us today. And I know you've got to run.
Let me throw two more things out there. One has been mentioned, and that's embryo models, or synthetic embryos, or symbrios, or they've got like 8 names. They're really interesting because we don't know what the hell they are. And we need to figure out what they are for things like laws that ban embryo testing after 14 days. there's no federal law in the US that bans that, but there are laws in some states and there are laws in many countries, is a embryo model made from iPSCs, is that an embryo for purposes of those laws? It's an area where I actually argue we need scientific research to answer that ethical question. I think, I'm a simple man, and if it walks like a duck and quacks like a duck and flies like a duck, I'm going to call it a duck. So if an embryo model could turn into a living human baby, I'm going to call it an embryo. We could do that research, except that it would be grossly unethical to try to take an embryo model, put it in a woman, and see what happens. But we could do that research in non-human primates and see if you get monkey pups out of rhesus macaque model embryos. It's not perfect evidence about what would happen with humans, but it's some evidence. So, I mean, that's to me a nice example of where science and ethics could work together. But you got to find a scientist who wants to do it, wants to work with monkeys, which is complicated for all sorts of reasons, and can get a grant to do it. When it may not lead to a great scientific advance, but it helps ethicists, or it helps society, it helps regulators. Another thing I want to talk about, just briefly, the weirdest thing I've been an author on, I think, came out last month in the MIT Tech Review, a short piece with Hiro Nakauchi, a stem cell scientist at Stanford, and one of his postdocs or grad students, Karsten Charlesworth, about bodyoids. Hiro has this idea of making animals either with no brains or with brains that aren't capable of sentience or consciousness for laboratory animal use because they'd feel no pain, maybe for meat because they'd feel no pain, maybe make humans that way for organ transplants. This is sort of the ickiest thing I've ever written about, but it's the kind of problem I really love. I've got a strong visceral reaction toward it, but I haven't come up with any really great reasons to justify that reaction. Those are the kinds of problems I love, where there's a strong reaction and trying to figure out why it is and whether I should have that reaction or not. But I think bodyoids have huge scientific questions. Could we do it at all? Could you have a living animal that doesn't have a brain? Leaving aside occasional politicians who are some proof one way or the other, would it be a decent model for an animal that has a brain? I don't know. But I think it's the sort of thing we should start thinking about, because there are some pluses and minuses, just like we should start thinking about ectogenesis, embryo models, and all sorts of really fascinating issues. This is the best time ever to be alive and interested in these questions, because compared to 30 years ago when I started in this general area, we know infinitely more. And 30 years from now, they're going to look back and say, oh, those fools in 2025, they thought they knew something. So things are changing fast, and it's fun to be along for the ride.
Oh man, that's for sure. Although I just a note for just to be clear and get it out there, the bodyoids were scooped by The Island with Ewan McGregor and Scarlett Johansson. And if anybody's seen The Island, spoiler alert, the twist is they couldn't really do it in the end. The people did have brains. They were clones, and they were killing them all along. But that's just Hollywood.
And The Island has been scooped by Lois McMaster Bujold, great science fiction author, with brain transplants from clones that are grown for that purpose. Science fiction is a wonderful source of ways to think about possible futures. It does have this problem, though. Fiction needs drama. And drama needs conflict, and dystopias are much more dramatic than utopias. And both of those are more dramatic than what we usually do in this world, which is muddle through. Humans aren't very good at doing anything perfectly for good or perfectly for evil, but we're pretty good at muddling through. And so here's to muddling through. My goal is to help us muddle through successfully and avoid catastrophes.
Wow, I couldn't have said that better. That's beautiful. We still got to get just a final note,
Sure.
just a couple final questions for our listeners to get a bit behind the curtain here. First one is, if you could answer any single scientific question, regardless of your expertise or chosen field, what would that be, Hank?
I'm going to cheat and make it a medical cure.
That's
I would
fine.
cure schizophrenia.
Oh man.
It affects 0.8% of the world's adult population. It is enormously disabling, disruptive to not only the patient, but to the family members. And it's so cruel because it almost always strikes people in the bloom of their early adulthood,
Hmm.
in their late teens or in their 20s. And I know schizophrenics, and it's a very hard life. It's an evil, cruel disease. If I could snap my fingers and fix one, it wouldn't save the most lives, but I think it would be what I would pick to fix.
Yeah, well, you mentioned it earlier. The brain, the complexity of the brain makes
Yeah.
disease mechanism there so elusive, but such a wide scope. I agree with you there. Finally, if you were not a bioethicist and deep thinker on the subject, what do you think you'd be doing with your life?
Professional tennis player.
Of course.
No, I know. I love playing tennis in the worst possible way, which is how I play tennis. I don't know. I've given some thought to what I'd like to do in my next lives. And I still kind of wonder what I'm going to do when I grow up, because I tend to have sort of an academic hyperactivity syndrome. I do genetics and neuroscience and stem cells and assisted reproduction and bright, shiny objects. I'm not really deep. I go wide and I hope not too shallow. Part of me would like to be a geophysicist who studies the evolution of the Earth's surface and the Earth over the last 4.55 billion years. Part of me would like to be a historical linguist who studies the evolution of languages over the last who knows how many years. Part of me would like to be a cosmologist who studies the evolution of stars and galaxies and the universe. So there is a common theme here, and I could be any kind of historian. I like to see how things change over time. And biology has certainly been a good field for that. And for better or for worse, that's not going to change in the rest of my lifetime or yours.
Well, you're talking about very different time scales here, from the evolution of language to the cosmos. But I'm glad we've got you here in our dimension, looking into the questions that need delving. And if you have looked broad and shallow, then I don't want to go deep, man. I think I might drown, because this has been a really, I would argue, a deep dive across the board for Arun and I and our listeners. So we appreciate it greatly. Thank you so much for joining.
Well, most importantly for me, it's been fun. Thank you, and good luck with the show.
All right, you guys, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at StemCell Podcast, Blue Sky at StemCellPodcast.com, or via email at info@StemCellPodcast.com with feedback or to suggest guests. This has been a real stirring episode. Get us thinking, hopefully got you thinking too. Next episode, we have the Honorable Sarah Teichman. Really looking forward to that. Until then, thank you guys so much for listening.